Good morning, everybody. Thank you for tuning into my talk today. My name is Osvaldo Rivera, and I am a cancer biology student at the University of Pennsylvania. I am excited to tell you today about our discovery of how splicing variations contribute to the functional dysregulation of genes associated with acute myeloid leukemia. Acute myeloid leukemia, or AML, is an aggressive type of hematologic cancer. As shown in this schematic, AML is a highly heterogeneous disease characterized by the proliferation of a poorly differentiated myeloid cell population. There are around 70 genes that have been found to be commonly mutated in AML. However, the genetic basis of this disease remains largely unclear, as mutations in individual genes are lowly penetrant and express complex patterns of co-occurrence and mutual exclusivity. Mutations in the FLT3 tyrosine kinase are the most common, but even these are present in only around 30% of AML patients. Mutations in other genes are even less common, with most occurring in less than 10% of patients. While a few splicing factors are among the so-called commonly mutated genes in AML, these are also present in less than 10% of patients, and no common patterns of splicing dysregulation has been associated with these mutations. Importantly, however, mutations are not the only way to dysregulate a gene. In particular, as most of you may know, alternative pre-mRNA splicing is a gene regulation mechanism capable of disrupting or altering protein function. Therefore, we sought to investigate if altered pre-mRNA splicing patterns increase the proportion of patients with loss of protein function for a particular gene. To address this question, we constructed a pilot cohort of 29 AML samples. We collected samples via apheresis and performed RNA-Seq on samples that were more than 90% viable AML blasts. We analyzed the RNA-Seq data using our splicing quantification algorithm called MAJIQ, which quantifies both standard splicing events as well as those that are complex or unannotated. I want to emphasize that our goal was to determine if splicing adds an additional layer of dysregulation to those genes already associated with AML. Therefore, we focused our investigation on around 70 genes that have previously been associated with sporadic cases of AML. Amongst these 70 genes, the MAJIQ pipeline quantified a total of 506 alternative splicing events across the PENN AML cohort. For my analysis, I am defining a splicing event as any case of altered splicing connections amongst a set of exons, as can be seen in these examples. Most of these splicing events exhibit low variability among samples, which is expected given the general need for splicing to be conserved as a consistent step in gene regulation. However, we do find 40 splicing events that we define as highly variable based on having a variability greater than one standard deviation above the mean. To determine if this is a unique feature of our dataset or representative of a pattern within AML, we next assess these 40 highly variable splicing modules in the larger publicly available BEAT-AML cohort as well as a cohort of healthy myeloid cells. Strikingly, we see that the variance of these 40 modules is significantly higher in both AML cohorts compared to healthy myeloid cells. In this violin plot, we have the sample groups versus the variance of psi for each of the 40 splicing modules. Having found the splicing variability, we first wanted to ask, are any of these splicing modules co-regulated? Indeed, we find that a subset of these variable splicing modules is highly correlated, suggesting that these modules may be co-regulated. What I'm showing here is a 40 by 40 pairwise clustering heat map of splicing modules in the PENN cohort. This heat map is colored and sorted based on the absolute Spearman correlations between splicing modules, such that yellow or orange indicates a strong correlation between the splicing pattern of two modules. As you can see, cluster 1, shown here as C1, represents the cluster of highly correlated splicing modules that includes 23 of the 40 modules. Importantly, we see that the co-regulation that we are observing in the PENN AML cohort is also present in BEAT-AML cohort and absent in the CD34 cohort. To address the potential functional relevance of these splicing modules, we first wanted to ask if any of these modules can phenocopy a genetic mutation. We were struck by the fact that the most highly variable splicing event in the PENN cohort is precisely in this cluster C1 and corresponds to a novel splicing event in EZH2. The gene EZH2 encodes for the catalytic subunit of the PRC2 repressive complex, which regulates gene expression and cell cycle progression. Loss of function mutations in EZH2 have been shown to block myeloid differentiation, suggesting a direct link between loss of EZH2 protein expression and AML pathology. Strikingly, we find that a subset of patients with both the PENN and BEAT-AML cohorts exhibit a high amount of skipping of EZH2 exon 11-12, shown here as low psi, and that this level of skipping is not observed in normal cells. To our knowledge, this splicing event has not been previously described, presumably as it is a complex event that is not easily quantified by many of the other splicing analysis algorithms. Importantly, this splicing event leads to a frame shift that leads to the introduction of a premature termination codon, or a PTC, which is capable of disrupting EZH2 protein function. Using RNA and protein samples from the PENN AML patients, we have confirmed our prediction in which we find loss of EZH2 protein expression in those samples with a high amount of skipping of EZH2 exon 11 and 12. As a side note, we also find another splicing module in EZH2, this one representing the inclusion of cryptic exon 9b. This particular splicing event has been described in the literature and has been experimentally shown by the groups of Robert Bradley and Omar Abdel-Wahab and others that inclusion of cryptic exon 9b is targeted for nonsense-mediated decay and functionally disrupts EZH2 protein expression. Critically, this data demonstrates that while only 14% of PENN AML patients, or 4 out of the 29, have detectable mutations in EZH2, an additional 8 patients are predicted to have at least a twofold loss of EZH2 expression via skippings of exons 11 and 12 or inclusion of exon 9b. Therefore, consistent with our initial hypothesis, accounting for splicing triples the number of AML patients with reduced EZH2 function relative to that predicted from cis-mutations alone. We next wanted to investigate possible mechanisms for this aberrant splicing. Studies describing EZH2 cryptic exon 9b inclusion suggest that it is mediated by genetic mutations in splice factor SRSF2 and/or U2AF1. Consistently, we observed that many of the PENN AML patients that have high inclusion of EZH2 exon 9b also have mutations in SRSF2. However, our analysis cannot establish any association between a particular splice factor mutation and the occurrence of EZH2 exon 11-12 skipping. Since we don't observe any known AML mutations that correlate with the EZH2 exon 11 and 12 skipping or any of the other correlated events in cluster C1, we asked if we could leverage the apparent co-regulation of these events to predict regulatory mechanisms. We know that splicing is regulated by RNA binding proteins binding to select sequence regulatory elements in the RNA. When we look at sequences around the junctions involving these splicing modules, we find an enrichment for polypurine motifs. Interestingly, we also find strong correlation between expression of polypurine binder SRSF1 and most of the 23 co-regulated modules from cluster C1. A similar but weaker trend was also observed for HNRNPF, but not for any of the other purine-rich binding factors. We have not been able to directly demonstrate that decreasing expression of SRSF1 is sufficient to drive the splicing changes we observed in the AML patients. However, our data does suggest potential regulatory factors that may drive, perhaps in concert with other alterations in AML, at least some of the variability in splicing across AML patients. Finally, up to this point, we've discussed splicing data on genes that have been associated only with sporadic cases of AML. Moving forward, we decided we wanted to look at another set of genes that in this case have been recently associated with familial cases of AML. Interestingly, when we run our splicing analysis pipeline, we see a similar trend in which a subset of genes are expressing highly variable splicing modules. In this data, we were particularly struck by the variable inclusion of a cryptic poison exon in the gene encoding DHX34. DHX34 is involved in activating nonsense-mediated decay via phosphorylation of UPF1. Inclusion of exon 12B, which introduces a PTC, is significantly higher in both AML cohorts when compared to CD34 normal myeloid cells. Inclusion of exon 12B is expected to lead to a loss of DHX34 protein and consequently a downregulation of the nonsense-mediated decay pathway. Therefore, we looked at the expression of genes targeted by DHX34 for NMD to see if there's evidence of dysregulation of NMD in these patients. Shown here are two plots where we can see the correlation between DHX34 exon 12B inclusion and the expression of genes PARP6 and WAM. These two genes are part of a subset of NMD target genes for which expression is significantly correlated with the DHX34 exon 12B inclusion in both the PENN and BEAT-AML cohorts, as shown in these plot of the Pearson R coefficient values associated with each cohort. These correlations maintain a sense of consistent directionality between AML cohorts. In conclusion, I hope that today I was able to convey the importance of splicing variations as a key component of gene dysregulation and disease etiology. We found that splicing variations are able to disrupt the coding potential of AML-related genes and therefore increase the proportion of loss-of-function aberrations that can be found in a particular AML cohort. We found that many of these splicing variations are correlated with each other, suggesting co-regulation and generating new questions about how we can leverage the use of global splicing patterns to designate and design novel therapeutic interventions. With that, I'd like to thank my advisors, Dr. Joseph Barrage and Kristen Lynch, as well as our collaborators and our funding sources. Please feel free to email me with any questions, and thank you for listening.